Hello students, welcome to the live session of Immunology course of NPTEL. Hello, I am Shurasri Pal. I am a PMRF scholar at IIT Kharagpur and I will be discussing some of the problems in these live sessions, some of the problems that you will face in Immunology course. We will be discussing these questions in this live session. So the first problem, so today is introduction, and in this session we will be discussing some basic questions which are not related to immunology, but this will be basic to studying immunology. So these are some of the fundamental questions. So the first question that you can see here on the screen is that in SDS page, a protein sample is treated with sodium dodecyl sulfate or SDS. This is a very common non-ionic detergent used. And what is its function? What is its function? So impart negative charge to the protein, make the protein become positively charged, change the pI of the protein, or change the protein density. So this SDS molecule is a non-ionic detergent, and the use of SDS in case of SDS page is that the proteins samples are run in gel. So when they are run in this polyacrylamide gel, what is the basis of their separation? What is the basis of separation of different protein samples in the polyacrylamide gel? And the basis of separation is that the molecular weight of the protein is the determinant factor by which how you can separate different proteins in the sample. And for separating proteins according to their molecular weight, what we need to do is we need to denature the proteins. And what SDS here, sodium dodecyl sulfate, does is that it denatures the protein. So suppose this is a protein molecule in its three-dimensional form, and so this protein molecule will have some positive and negative charges on the surface. But if this three-dimensional form, like we cannot run this three-dimensional form of the protein in the gel, if it is run in this form, then they will not be separated according to their molecular weight, but they will be separated according to their shape and size, so which is not desired in this case. So what SDS does here is that it linearizes the protein, and not only that, it imparts uniform negative charge to this linear protein molecule. And this is very important, uniform negative charge. This is important because in polyacrylamide gel electrophoresis, we will see that we have two electrodes, a positive electrode and a negative electrode. And the protein molecules which will be negatively charged by the application of SDS, they will move towards the positive electrode. So negatively charged protein molecules will move towards the positively charged electrode, and by the application of electric voltage, and which is how we will run the protein molecules in the gel, and then how far they will migrate will be determined by their molecular weight. So the use of SDS here, we can see that it not only denatures the protein molecule, but also uniformly imparts negative charge to the protein. So you can guess this option A, impart negative charge to the protein, will be the right solution here. So this is the solution. SDS imparts negative charge to the protein. Okay, now coming to the next question that we will discuss in today's session. The enzyme found in retroviruses, which is required for the synthesis of DNA from RNA, is DNA polymerase 3, RNA polymerase, ribonuclease, or reverse transcriptase. So retroviruses are the type of viruses which do not have DNA as their genetic material. So the genetic material of retroviruses is RNA or ribonucleic acid. Now what happens when these retroviruses infect their respective hosts? So they need to produce the viral particles, and so the virus needs to replicate in the host, and for that the first thing that should be occurring is that it should copy its genetic material, or it should replicate its genetic material. And we know that if RNA is the genetic material, then it has to convert it into DNA first. So for converting RNA to DNA, we need RNA dependent DNA polymerase, which can convert RNA to DNA, and this enzyme is called reverse transcriptase. Why is it called reverse transcriptase? Because when the cell produces RNA from DNA, this process is called transcription, right? And the process is mediated by the enzyme RNA polymerase. Production of RNA from DNA is called transcription, and it is mediated by RNA polymerase. However, when this reverse happens, so RNA to DNA, when this reverse process occurs, this is called reverse transcription, and the enzyme is reverse transcriptase. Okay, so here option D is the correct answer, correct solution. Okay, we will also discuss some of your doubts if you have in this portion, or we will move on to the next question, which is, which of the following is not true for chloroplast and mitochondria? So here we have to focus on this terminology, which is not true. This is very important. So always we have to look out for the options or the statements which are not true. So first of all, the first statement is both have an endosymbiotic bacterial origin. So endosymbiotic bacterial origin means, according to the theory of according to the theory of endosymbiosis, we know that chloroplast and mitochondria, these were possibly, or we hypothesize them as to the prokaryotic cells, which were later incorporated within eukaryotic cells, and existed in a symbiotic condition with the eukaryotic cells. So this is true. This statement is true, that both have an endosymbiotic bacterial origin, right? Now next is both are associated with energy production. Now we know chloroplast is the site for photosynthesis, and mitochondria is the site for cellular respiration. So both photosynthesis and cellular respiration, they are both important for energy production. So photosynthesis traps the energy of the photon, which is from the sunlight, and in respiration we utilize that energy for our so we produce the energy units which will be used in our cell, like the ATP. So obviously both are associated with energy production, so this is also a true statement. Next is number C. The chloroplast is found in plant cells while mitochondria in both plant and animal cells. So this is also true, obviously. Next, coming to the last option, which, since by the hypothesis of nullifying, we can say that since these are true, so obviously the last option will be false or not true, but we will still verify it. So it says chloroplast consumes oxygen, but mitochondria produces oxygen. So this statement here is not true, as we know that photosynthesis is the process where in the Hill reaction oxygen is produced in the chloroplast. So chloroplast consumes oxygen is a wrong statement, while mitochondria produces oxygen, that is also a wrong statement, because in mitochondria cellular respiration takes place and cellular respiration uses oxygen. So this statement is not true. So we can say here that the correct option will be option D, solution D. Well, now coming to the next question, which is, which property of water supports life in polar region? Water is a universal solvent. We know that this statement is true. Water maintains an equal level. Expansion of water upon freezing. This is also a statement which is true, and the last option is all of the above. Now, what happens in polar region? So we know that in the water bodies which exist in polar region, they often freeze. So the lakes and the water bodies, they have a layer of ice which forms on the surface. However, the aquatic animals of that region or of that water bodies, still they survive in this kind of frozen lakes. So how does that happen? This happens because of the phenomena called anomalous expansion of water. So we know that water, the density of water is highest at 4 degrees centigrade, and when the water freezes, its temperature is 0 degrees centigrade, right? So when water goes down from 4 degrees C to 0 degrees C, it becomes less dense. So density decreases when it goes from 4 degrees C to 0 degrees C. So because of which it expands in volume. So we know that the volume of same amount of water is lesser than the volume of same amount of ice. So when it freezes, it expands in volume, and because of which what happens is that, so water not only it is it expands in volume, but also because it has less density, it comes up to the surface of the water bodies, and this so this ice, the frozen water, comes to the surface of the water bodies, and what happens is that it covers the entire surface, and we know that ice is not a good conductor of heat, because of which so when the surface of the lake freezes, the inside the water which is there below the surface of this ice does not freeze. So as we go down, the temperature of the water can exist. So the temperature can be still 4 degrees centigrade, and in which in this 4 degrees centigrade, so the aquatic animals can survive. So as we can understand from here, that expansion, because of this anomalous expansion of water upon freezing, water in the polar regions, in the frozen lakes, still supports life or supports the life of aquatic organisms. So of course here this is the correct option, expansion of water upon freezing. Okay, and coming to the last question that we will discuss in this session. DNA is not the genetic material of option A protozoa, option B bacteria, option C blue-green algae, and option D retrovirus. So this is something we discussed in one of our previous questions also. Retrovirus will be the correct option because retrovirus has this genetic material which I mentioned earlier, it's RNA. So DNA is not the genetic material of retrovirus. So that is the correct solution here. So all right, thank you for attending this session. I'll be terminating this session here. Thank you for joining.